Okay, moving on to lipids. So, lipids are a diverse group of hydrophobic molecules, and we already know what hydrophobic is, but let's continue on. Alright, so lipids are the one class of large biological molecules that do not form polymers. So, the unifying feature of lipids is they have little or no affinity for water. And lipids are hydrophobic, and this is because they consist mostly of hydrocarbons, which form nonpolar covalent bonds. So we talked about this briefly last week. We have carbons that are polar, so because water is polar, they're not going to be soluble in water, so they don't dissolve. But the most biologically important lipids are fats, phospholipids, and steroids. So, beginning with fats. Fats are constructed from two types of smaller molecules, glycerol and fatty acids. Glycerol is a 3-carbon alcohol with a hydroxyl group attached to each carbon, which are lots of words, but we'll get onto that in a moment. And a fatty acid consists of a carbonyl group attached to a long carbon skeleton. So let's take a look. Okay, so here we have a glycerol, so the 3-carbon alcohol with the hydroxyl group. So here's the 3 carbons in the glycerol in the gray, and then it's attached to a hydroxyl group, so that's the OH there. And so because you have that OH there, and then the fatty acid consists of a carbonyl group, which is this. Well, it's the carbon and the oxygen, double bond to the oxygen, and then the OH group over there, that's the carbonyl. So then because you have hydroxyl in the carbonyl group, you can do the dehydration reaction, take that out, and then you can join that together with the ester linkage. So, but because you have these 3 carbons with the 3 hydroxyl groups, you're able to attach 3 fatty acids to it, and that makes a triacylglycerol, which is a fat molecule, most common fat molecule. So we have glycerol, fatty acid, and when then you complete that, you have a triacylglycerol. Okay, so that's what consists of a fat. Okay. So fats separate from water because water molecules form hydrogen bonds with each other and exclude the fats. In a fat, 3 fatty acids are joined to glycerol by an ester linkage, making it a triacylglycerol or triglyceride, so we saw before. Oops. It's like right here. And the fatty acids can vary in length, so that would be the number of carbons. So you see how many carbons you have along in the yellow. You can make it longer or shorter. And the number of locations of double bonds. So we'll talk about that in a second. So saturated fatty acids have the maximum number of hydrogen atoms possible and no double bonds. And so unsaturated fatty acids have 1 or more double bonds. So what this means, first of all, you probably heard of saturated and unsaturated fatty acids like in a nutrition label, or when you talk about like fast food or something. Like, what exactly does that mean? So saturated fatty acids means that you see all the carbons here in the yellow. It means, and all the hydrogens that are attached to each of the carbons. You can introduce a double bond, and so you can introduce double bonds between the 2 carbons, and then you would take out some hydrogens. But when you introduce a double bond, you can introduce a kink. Here's another picture. So a kink means that, well, I'll explain in a second. Anyways, saturated fat means it's straight, no double bonds. Then unsaturated fat means that you have 1 or more double bonds. You can kind of see it really tiny in the bottom thing, but that double bond causes a kink. So here's an example of that. But one reason that, let's talk about this first. Alright, so fats are made from saturated fatty acids and called saturated fats, and these are solid at room temperature. Most animal fats are saturated, and then fats made from unsaturated fatty acids are called unsaturated fats or oils, and are liquid at room temperature. So think olive oil, like in the first picture, and then plant fats and fish fats are usually unsaturated. So think oil for unsaturated fat, and like solid, like butter, or like when you grill a hamburger or something, the grease eventually becomes solid. But that wouldn't be so for like a plant oil or a fish oil. So a diet rich in saturated fats may contribute to cardiovascular disease through plaque deposits. And so this is one reason, is because, well, I'll explain in a second. So hydrogenation is the process of converting unsaturated fats, or the kinked ones, to saturated fats, which are straight, by adding hydrogen. And hydrogenating vegetable oils also creates unsaturated fats with trans double bonds. So these trans double bonds will contribute more than saturated fats to cardiovascular disease. So what trans double bonds means is basically you just make a double bond that, instead of making a kink or making it curved where it makes a double bond, it makes it straight again. So this particular double bond is a cis double bond, so that means they're going to go in two opposite directions, but trans just means it flips it over and then the molecule keeps going straight. So one reason one way you can think about this is in saturated fats, one reason why they're solid at room temperature is because of the structure. I'm talking about kind of structure and function. So these flat saturated fats can all pack together, and so that's what makes them solid. So like one reason you don't necessarily want to get processed peanut butter is because they convert the unsaturated fats to saturated fats, so that the peanut butter isn't as oily as when it is at room temperature. So I don't know if you guys have gotten like natural peanut butter or something before from the store. So if you've got natural peanut butter, you'll see there'll be like an inch or so of oil at the top before you mix it up. And so that's the way peanut butter naturally is. But with processed peanut butter, they change those oils from the unsaturated fats to saturated fats, so that everything mixes together and it's not oily and it's more easy to spread. Unfortunately, when you have those flat structures, when you integrate those into in your cells, or they get into your bloodstream, your cell walls, so like what we'll talk about in a second, you guys talked about the phospholipid bilayer, but the kinks, some of those are straight and some of them are crooked, but the crooked ones help the phospholipid bilayer to be more fluid and not be rigid. So that's one reason why it's important for your health not to have too many saturated fats, because you can have fat and your cells can pack together, and then you just have an unhealthy amount of straight phospholipids so in your cells, and then also having these can also form plaque in your blood vessels, and so those build up, those pack together, and your blood vessels accumulate these saturated fats, and they become rigid and hard, and so that's not good either for your blood circulation. That can cause cardiovascular problems. So structure and function in saturated fats. Alright, so the major function of fats is energy storage, and then animals and other mammals store their fat in adipose cells. And adipose tissue also cushions vital organs and insulates the body. Alright, moving on to phospholipids. Phospholipids are two fatty acids and a phosphate group that are attached to glycerol. So the two fatty acid tails are hydrophobic, but the phosphate group and its attachments form a hydrophilic or water-loving head. So here's a picture. Here's the hydrophilic head made out of the glycerol and the phosphate groups. So here's the phosphate and here's the glycerol. Then you have the two hydrophobic tails of the fatty acids. Here's a more detailed picture. So when phospholipids are added to water, they self-assemble into a bilayer with the hydrophobic tails pointing in towards the interior, and the structure of phospholipids results in a bilayer arrangement found in cell membranes. And phospholipids are the major component of all cell membranes. So we can kind of see a picture here. So picture you have these hydrophobic tails that want to be away from water, and the hydrophilic tails, or hydrophilic heads, that want to be towards water. So the hydrophobic tails kind of are grouped together. They want to protect themselves from the water, and then they have the hydrophilic heads that want to be near the water, so it all kind of works out. The hydrophilic tails kind of band together, and then the hydrophilic heads point out towards the water, and you get this bilayer, and it all seems to work out. Alright, so steroids are lipids that are characterized by a carbon skeleton consisting of 4 fused rings, like this. So each of these hexagons and one pentagon is made out of carbons. And cholesterol is an important steroid and is a component of animal cell membranes. And it is another structure that kind of helps break up and yet stabilize the cell membrane. It helps it to stay more fluid at certain temperatures, but also helps it to keep from freezing and becoming rigid at freezing temperatures. However, unfortunately, if you have too much cholesterol, that's why it's essential in animals, but if you have too much cholesterol, it can also contribute to cardiovascular disease. So there's a picture of cholesterol. And so concludes our lecture on lipids.